Okay, so in question 10.13 we are drawing a dinucleotide, and we're going to draw its complement too. So we're going to draw an A and G, and then on the other side we're going to draw a T and C, and we're going to show those nucleotides bound to one another. And let's just focus on the nitrogenous bases first. Okay, so let's start with adenine. And adenine should look something like this. Don't start with a nitrogen, start with a carbon. Here we go. You can start with a nitrogen if you want. Carbon, nitrogen, carbon, nitrogen. Okay, we have some double bonds here and here, and I'm going to draw, this is adenine, so we're going to have a nitrogen attached up there. I ran out of room, so I have to kind of put that angle in there. We're going to have a double bond here and double bonded oxygen there. And this one right here will be attached to the sugar. Okay, I think everything looks good here. Another hydrogen here. Okay, everything looks pretty good here. I think we're missing a double bond there. Okay, everything looks good. So here's our adenine. Now below that we will draw the guanine, right? So it's a purine. It looks a lot like adenine. So I'm going to draw these bonds in here, these atoms. Okay, looking good. Well, it doesn't look that good, but all the bonds are in the right place. I mean, all the atoms are in the right place at least. So let's, and the next one, so this is guanine, so this is going to have a double bonded oxygen there. It's going to have a hydrogen here and a double bonded, what is this, guanine is going to have a amino group down here, right? Let me check on that. Yes. Okay, so it's going to look something like that. And where are all the double bonds? So we're going to put one here. We are going to put one here. And we already have one over there. Okay. So this is going to be our guanine. So all the carbons should have 4 bonds, all the nitrogens 3 bonds. And I think we have that taken care of. Okay, this one right here. Going on with that one? Oh yeah, this one is attached to the sugar. So sometimes it's good to look through the molecule and make sure all the nitrogens have 3 bonds, carbons have 4 bonds. Okay, so we have our adenine and our guanine. So now let's draw in the thymine and the cytosine. Okay, so the thymine will go over here, and I'm going to have to draw it in a different direction than what I'm used to drawing, or how I'm used to drawing it. I have to put the nitrogens on this side over here. So something like this would be good. And this is thymine, so we're going to have a methyl group over here, hydroxyl up here, and a hydroxyl, not a hydroxyl, but a oxygen, double bonded oxygen to a carbon right there. Okay, double bonded oxygen to that carbon there, and we are going to have a hydrogen in here. And this is our thymine, and this one right here is going to be bound to the sugar. To check our bonds, every carbon has 4 bonds now. This one's double bonded here. Nitrogens have 3 bonds. Okay, looks good. Okay, so now our hydrogen bonds, I'll draw these in red. So one of these hydrogens of this nitrogen up here is going to form a hydrogen bond with this oxygen over here, and then this this nitrogen right here has a kind of a slightly negative charge with it, and it's going to form a bond, hydrogen bond with that hydrogen of that nitrogen right there. So those are our two hydrogen bonds. It's not the neatest diagram, but at least we see that this nitrogen hydrogen bonded to this hydrogen of that nitrogen, and this hydrogen up here is hydrogen bonded with this oxygen over here. Okay, let me check to make sure that's right. Yeah, looks pretty good. Looks good to me. Okay, excellent. So let's go down here and let's draw in the cytosine. Okay, so let's draw the cytosine in here next. Okay, so I'm going to start with the nitrogen down here. Carbon, nitrogen, carbon, carbon, carbon. And we're going to have an NH2 right here. I'm going to draw it like that. Double bond here. Double bonded oxygen here. And a double bond here. Each one of these is going to get a hydrogen. This is going to be attached to the sugar. And okay, that looks pretty good. So we are going to see a hydrogen bond form between that oxygen and one of these hydrogens here. This hydrogen is going to form a hydrogen bond with this nitrogen over here, and this one of these hydrogens will form a hydrogen bond with this oxygen right there. So now we need to draw these attached to the sugars, and that's going to be really difficult, especially with this small space I have to draw the structure. So let's see if I can, shrink this down a little bit. I'm going to copy that, and let's see, I can paste it, and let's see if I can shrink it. No. Like that. Okay, it doesn't look that great, but I just kind of want to draw the sugars around these so we get an idea of how you would draw a structure like this, or what it should look like at least when we have the sugars attached to these. So, okay. So how would I draw the sugars here? Let's see. I'm going to take the pen and I am going to draw the sugars over here. I'm going to start, let's see, I'll cross that out and just kind of draw a long bond there. So this isn't, these bonds are obviously too long, but I just kind of want to show how the atoms are all linked together. So this will be one of the 1' carbon of the sugar, oxygen, carbon, carbon, carbon here. Here's the 5' carbon, and this will be attached to an oxygen, which is attached to a phosphate, which is attached to another oxygen, oxygen, oxygen. Okay, now our molecule's starting there, so I'm not going to draw anything else up there. We have 2 hydrogens here, 1 hydrogen here, right? 1, 2, 3, 4. Here we're going to have an oxygen and a hydrogen, and I have a phone call. You're going to have to wait on that. Okay, sorry about that. I had to answer that call. Okay, where was I? Yes, I'm attaching these sugars to these bases. So, okay, we're going to have these are DNA bases, so we're going to have a hydrogen here, a hydrogen up here, 1, 2, 3, 4. Okay, everything looks good. Now this is where we're going to have our phosphate, right? So double bond here, oxygen here, and this is going to be attached to the 5' carbon of this sugar right here. So let's draw that in here. Carbon, carbon, carbon. Oh, that's wrong. Let me scroll down a bit. There's the carbons we want. Okay, now this is the bond that's forming up there. Okay, so we have all the bonds formed correctly. I can put some hydrogens in here, 1 here, 2 here. This is going to be attached to a hydroxyl, or a phosphate group. Another phosphodiester linkage will form there if there's another nucleotide attached there. Remember, that's the 3' carbon. So a hydrogen, hydrogen, and 2 hydrogens right here. Okay, so here we go. We're going from the 5' to the 3' direction. This is the 5' carbon, this is the 3' carbon, this is the 5' carbon. Okay, that's great. Now, we have to draw the sugars on the other side also. So let's see if we can do that. And they have to be sort of going in the reverse direction, right? And I've never drawn these upside down. I'm going to give it a try. Okay. So let me see. Where's the best place to start this? How about... Okay, so this is going to be attached to the sugar here. So how about like this? Okay, carbon, carbon, the oxygen's here, carbon, carbon. So here we're going 1', 2', 3', 4', and then the 5' carbon will be right here. H2 attached to the phosphate. Okay, so I think that looks pretty good. How about we need some other hydrogens in here, right? How about 1 here? We only have 2 bonds here, so we're going to put 2 hydrogens in there to take up those other 2 bonds to the carbon. Hydrogen right here, and this one is where we're going to have our 3' carbon, right? This is where we'll have our phosphate, phosphodiester linkage, and we need 1 hydrogen there too. So oxygen, phosphate, oxygen, oxygen. Now, and this is attached to the 5' carbon of this sugar right here. We're running out of space, so this is not how the carbons would be. This is how they'd be linked, how the atoms would be linked to one another, but not necessarily how they'd be spaced from one another. So I am going to, what's the best way to do this? Okay, I'll just start right here with the 1' carbon, carbon, carbon there, carbon here, oxygen, and this is the 5' carbon right here. So 3' up here. This is going to have the hydroxyl, 2 hydrogens here, hydrogen here, and this is where we're attached to this nitrogen right here. Okay, I think that looks pretty good. Okay, so this is the 3'. This is the 5' over here, and you can see they're anti-parallel. These sugars right here are going in the 5' to the 3' direction, and these ones over here are going 5' and 3' in this direction. Okay, so that's how I would answer number 13.